Our speaker today is Jens Lagergren, who is a professor in computer science and computational biology at the Stockholm Bioinformatics Center. He has a broad collection of research interests in computational biology, from computational complexity proofs to probabilistic methods for data analysis. Today he will be telling us about one of his main research programs in a talk entitled Probabilistic Analysis of Gene Families with respect to gene duplication, gene loss, and lateral gene transfer. Go ahead.
Okay, thank you, Eric. So this is probabilistic analysis in the sense of Bayesian analysis using MCMC, but you can of course use it for other purposes also, like maximum likelihood. And I guess that most of you are familiar with these evolutionary events, but let me just start by introducing duplications. So let's see if I can do that. Okay. So here to the right in this cartoon, you have the obvious way of duplicating a gene or creating a new gene, that is by duplication of the DNA segment or retrotranscription. So you have one organism in which suddenly a gene is duplicated by some mechanism, and after that you have two copies of that gene. So you have two members of that gene family instead of one. The number of members have increased by one. To the left here, we have the other way of increasing the number of members in the gene family, namely by speciation. So we have one ancestral organism up here having one member of the gene family, let's call it G. And after this organism has gone through a speciation, we have two species, A and B, which each have a copy of that gene. So they both have a member of that gene family. So we're going from one to two members of this gene family. And this gives an obvious computational problem, namely when we have a gene tree, how do we classify the vertices of that gene tree into speciations and duplications? That is, how do we find out the events that correspond to the vertices that actually create, the event that created the split? And I'll be talking about this in terms of probabilistic models, and there are actually three parts of the talk. Each will be concerned with a generative probabilistic model. In the first one, we have the gene evolution model, which just contains duplications and losses, basically, or the evolutionary events, probabilistic evolutionary events we consider are duplications and losses. And here we get from a given species tree, a gene tree. And using that, we can turn around and ask, when we have a gene tree and a species tree, what is sort of the evolutionary, the reconciliation that is most likely to have given rise to these two? What is the scenario that is most likely to have given rise to this gene tree? And then there's two other models, namely GSR, which contains duplications, losses, and also sequence evolution. So in the first case, we will have to, if you use that, you have to reconstruct your gene tree using some standard method, and then you can reconcile it. But in the second case, you can do all of this inside one model, which is better than using a two-step process. And in the final part, I'll be talking about another model which contains duplications, gene duplications, lateral gene transfers, losses, and also sequence evolution. And the motivation for this, well, there's lots of motivations. And one is, or two are, that we want to reconstruct gene trees. And we also want to study the rates of these types of events. We want to study rates of duplications and lateral gene transfers. And this we can do now in the sense that we have implementations of algorithms, of efficient algorithms that we can use to do this. And later, in the sense that we have algorithms for these problems, but they're not fully implemented. is that we can do autology analysis with respect to GSR, so including sequence evolution. But we can also find where events have taken place and also where in a species tree. And then we have the harder problem, which also is a motivation, but it will come even later because we don't have any efficient algorithms for this right now. We're working to get it, but we don't have it yet. And what we want to do here is to reconstruct species trees. And this may seem like a lot of claims of things that we can do or things that we're aiming to do, but the reason for this is that we're doing probabilistic modeling. And here I have a picture that's supposed to illustrate GSR, one of the models. And what it does is that it relates several objects. It relates sequences for a gene family, a gene tree, an embedding of the gene tree into a species tree, which is called the reconciliation, and also a species tree with time of the edges, and finally rates. So actually, given a species tree and rates, this model will generate all the other things. But you can view a model like this like as an equation, basically, where you give some of the variables values. So you instantiate some of the variables, and the other ones, you sort of ask to resolve, or you want to find the possible values of. And what we do for GSR is, for example, that we have a species tree and we have sequences, and then we look for the posterior over rates, reconciliations, and gene trees. And actually, we marginalize this, so we integrate over rates and some over reconciliations, and that gives us posterior probabilities for gene trees. But of course, we can turn this around in any way, basically. And one of the things we can do if we want to get a species tree is that we take sequences for several gene families and ask for the most probable species tree to generate gene trees that then generate these sequences, or we can ask for the posterior distribution over species trees. Okay. So we have several articles on this, some older in conferences and several newer, basically from last year. There's one describing all efficient polynomial time algorithms, which is in the Journal of ACM. There's a paper in Systematic Biology where we show how to play around with things like this based on the gene evolution model, so where you only have a species tree generating a gene tree. And finally, we have a paper in PNAS where we describe how you also include sequence evolution for this. The stuff I will talk about for lateral gene transfers hasn't been published in the journal yet. I have a former student who included most of the theory in a thesis, and we will hopefully soon publish a paper on this. Okay, so let me start fairly basically and describe the type of reconciliation method that existed before we started to work on this. And that was basically only the most parsimonious reconciliation, MPR. And I'll describe how it works. So here we have a species tree in yellow, and over here we have a gene tree. And the species are A, B, and C. And the leaves of the gene tree are labeled with the species in which we have found the genes. So what happens when you do the most parsimonious reconciliation is basically that you put the extant genes on the extant species in which you found them. And I usually describe it as this, that you put all of the other vertices on up at the root or above the root of the species tree. And then you let the vertices of the gene tree fall as low as possible, basically so they straddle a vertex in the species tree. So the reason why we cannot put this particular gene lower than this is, of course, that if we would put it, say, here, then it would have, the gene would have a descendant in the species B, which is not a species that is and a descendant of the species in which we found this gene. So if we continue like this, both these vertices end up here, and then this guy is sort of supported by two previous vertices in the gene tree. And there, because of that, we put it on an edge, and according to this theory for the most parsimonious reconciliation, it must be a duplication. And we can continue like this and get this reconciliation, which is the most parsimonious one. So the most parsimonious reconciliation is defined like this, and it minimizes the number of duplications you can get. But there are lots of other reconciliations. In fact, if you put the extant genes on the species where you found them, and then you place vertices of the gene tree, either on where the MPR places it or an edge above, then you will get another reconciliation. So for instance, in this case, if we take this vertex and push it upwards, then it becomes a duplication. And this is another reconciliation. So you can, of course, compare reconciliations based on how many duplications you get, but then MPR is always the one to prefer. So we introduced the gene evolution model where there are two probabilistic events that affect genes, namely duplications and losses. And there's also speciations, but they affect them in a deterministic way. And here a gene tree evolves inside a species tree according to the following rules. First, a single gene starts at the root of the species tree, or it may start on an edge leading to this root. And then over any edge in the species tree, the gene tree evolves according to a linear birth-death process. When a gene reaches a species tree vertex, it is split into two independent processes, evolving down the two outgoing edges. And finally, losses are pruned, and also the vertices that become redundant when losses have been pruned are pruned recursively. And this is, from a mathematical point of view, is almost canonical. I mean, the birth-death process is sort of the first choice for something like this. And there are authors who have claimed that this is biologically sound. It's certainly intuitively appealing to many people. So here I have just an example of how this could happen. So we have a single gene that starts here in the root of the species tree. What happens to the right here is independent from what happens to the left here. So we can describe those things independently and say we may have a duplication on the right side, and both copies may survive down to the species down here. On the left, we may have a duplication followed by another duplication, and then all three copies may survive down here. And after this, both these guys, or each of these guys, start to evolve along this edge and along this edge, so independently there. And basically the same thing happens over here for these three vertices. And we may get something like this. And from this, we prune away the losses and get what I call a realization. So there's some important points to make here. First of all, we have time on the edges of the species tree. And also the birth-death process takes place in time. So any vertex or any duplication sort of happens at a particular time point when it, when we use this process. So by realization, I basically mean a reconciliation where we have exact time points for all duplications along the edges of the species tree. And the process gives realizations, which means that we also get probabilities for reconciliations. So the probability for any reconciliation that puts these 4 vertices as duplications like this, so all the 4 green guys here, we get that probability by integrating overall possible duplication times for these green vertices over the edges where they occur. So over this edge, over this edge, over this edge, and over this edge. So this is nice because now we can start to infer missing data based on this gene evolution model. So for instance, given a species tree and with time and a gene tree, we can ask for the posterior probability over reconciliations, rather than just picking one reconciliation that minimizes the number of duplications. Or we can, given a species tree, calculate or ask for the generation probability for a gene tree. And I just want to compare this with the situation we have for the gene evolution model with the situation for the coalescent. So I'm aware that coalescent is probably a better model than the gene evolution model when you want to model populations and lineage sorting. But when you want to model gene duplication, I would claim that the gene evolution is a better model because there's no real interpretation of the colliding event when you look at duplication of genes. And also in terms of efficient algorithms, we have a number of efficient algorithms for basically any probability you would like to calculate. So generation probability, the probability of a gene tree, the maximum likelihood reconciliation, we can compute that, the posterior over reconciliations, and we can sample from the posterior, and we can calculate orthology probabilities. But for the coalescent, you basically need a realization to be able to compute anything. But again, when instead you want to model lineage sorting, the coalescent is probably a much better model. Although it could be the case that the gene evolution model gives a decent approximation of the coalescent, and therefore is interesting also for lineage sorting. Okay, so let me give you an example of what we can do with this, or in general can do when we are able to calculate probabilities of this type. So here we have a reconciliation of a gene tree in black and a species tree in yellow. It is for gorilla and some close relatives. And actually, for various reasons, we know that this vertex here is a duplication. Actually, if we add data, the MPR will tell us that this vertex called 0 here is a duplication. But when we limit the data to what we see here, MPR actually claims that this vertex is a speciation. So sort of MPR is inconsistent here, depending on what data we give it. But when we use our probabilistic approach, we can of course look at other reconciliations, and we can consider the probabilities of those, and we can ask, is the probability is the probability for overall reconciliation that according to which this vertex is a speciation, is that higher than the probability overall reconciliation according to which this vertex is a duplication? And we can calculate that. I mean, the expression is the following, the mathematical expression. If we want the probability that the vertex is a speciation or a duplication, we can just sum over all reconciliations. because when we have a reconciliation, we know whether a vertex is a speciation or a duplication, and then multiply this with the probability of the reconciliation. And we can do this efficiently. Of course, in this expression, we have the rates. So, and in reality, we don't know the right rates, but there's a way around that, namely, you can integrate out the rates. So you can look at the posterior means over the rates. So in that way, you get the probability that the vertex is a speciation. But still, if you compare to the MPR, MPR only gives you one reconciliation that will tell you whether a vertex, according to it, is a duplication or a speciation. But here we get the probability, which means that we have the problem of picking a threshold. So, should we call a vertex a speciation when the probability that it is a speciation is higher than 0.6, or should we do it when it's higher than 0.8? And our solution to this is that, well, we can sample from we can sample the posterior over the rates, the duplication and loss rates, and based on that sample, we can create lots of synthetic instances, gene tree, species tree, and for each of those, we can try any threshold, and this basically gives us a sensitivity specificity curve, a rock plot, of the possible combinations of sensitivity and specificity that you can get for these particular birth and loss rates. And then a user, can pick any combination that he or she thinks is the best, and use that for the particular biological instance he or she wants to analyze. Okay, so that's all I'm going to say about the gene evolution model, and now I will turn to GSR, which contains also sequence evolution. Okay, now, as I said, from the gene evolution model, we get the gene tree and a realization. So we actually get the time point for any vertex of the gene tree when it occurred, which means that we get an ultrametric gene tree from the gene evolution model. And we could, of course, get a generative model that generates sequences simply by generating sequences according to our favorite model of sequence evolution on this ultrametric gene tree. But ultrametric gene trees are not so good, as we know, and it will turn out that had we used ultrametric gene trees, we would have been in trouble later on when we evaluate this. So what we do instead is that after the gene tree has been generated, it is ultrametric, then we perturb the times. Okay, we do this by generating a rate for each edge in the gene tree, and then we replace the time. Well, we multiply the time with the rate, and we get an edge length, and that's the length we use when we generate sequences in the gene tree, which we do, with any favorite sequence evolution model. Okay, and now we have a much richer model. So we can ask things like, given a species tree and a family of genes, so sequences aligned, what is the posterior of the gene trees? Or given a species tree with time, a gene tree, and a family of sequences, what is the posterior over different reconciliations? Or what is the posterior over combinations of gene trees and reconciliations? And finally, we can, in theory, we can, of course, ask for the posterior over a species tree when we have observed several gene families, although I already made a comment that this is hard to do in practice. Okay, so what is it that we can do? Well, one thing that we have implemented already now, and which is described in this PNAS paper, is how we construct gene trees. So in this case, the input is a species tree with time, and also a gene family that is sequences, aligned sequences. And the output is a distribution, posterior distribution over gene trees. In this case, so let's see now. Right, so this is an MCMC algorithm. It seems to work fine for reasonable sized instances, and I will tell you about some application of this. I'll just briefly sort of try to explain the inner machinery of this. So what we're doing is MCMC to compute the posterior after having observed the data, the sequences. And fortunately, we can do basically what MrBayes does, namely we can have a Markov chain where the states are gene trees, G, and lengths for the edges of the gene tree. And then parameters like our duplication and loss rate. I've hidden I've excluded the parameters for the sequence evolution. So we have states of this type. And what we want to compute for any combination like this is its posterior probability. And as usual, when doing MCMC, it doesn't matter that we cannot compute the posterior, which we actually want to compute, as long as we can compute ratios between posteriors. And we can, because as usual, we can use Bayes to express this left-hand side here as the right-hand side. And as usual, the probability for the data will disappear when we take the ratio between two probabilities of this type. And what we have up here is that, well, we have the probability for the data given the gene tree and the length of the edges, which is just the standard sequence evolution probability, what Felsenstein introduced to the field 81, and what's done in any, MrBayes, any program. And here we have the probability of the gene tree and the lengths given our parameters to the gene evolution model. And here we just have some, the prior for these parameters. So what we need to be able to compute is the probability for the gene tree and the lengths. Everything else is sort of trivial or already known. And we can compute this probability by integrating over different times, and we approximate this using a sum which can be computed using dynamic programming. Okay, so let me end the more mathematical part there and go over to comparing our results with the results obtained in this Nature article by Wapinski et al. What they consider is a number of fully sequenced yeast genomes, and they analyze all gene families from these yeast genomes. In order for us to be able to compare with them, we analyzed exactly the same gene families. And as most of you probably know, there has been a whole genome duplication in this yeast tree, exactly here where we have the star. And we will later on be concerned with recovering exactly this vertex. I mean, in the gene tree, it is a vertex, right? Because this part of the species tree from here to here is duplicated in any gene tree where there hasn't been losses after the whole genome duplication. So we'd be interested in recovering that vertex, but also this vertex here, which we call the pre-post whole genome duplication vertex. Okay. Now, when Wapinski et al. Used their method, which is called SYNERGI, they don't need any edge lengths, but we do need edge lengths or time dates on the species tree, and we use this the program described in this paper to date the species tree. Okay, here's the comparison with SYNERGI. Babinski et al. Classified their families into small, medium, large, and uniform. The uniform are families of the type where there's exactly one member of the gene family in each of the 17 species in the species tree. Okay. The small are simply gene families with up to, I think it's between 7 and 10 members. Medium is 10 to 17, but not including the uniform, and large is above 17. This is simply the number of gene families of different types, and as I said before, we use exactly the same gene families. Now, a difference between their approach and ours is that they pick out one single gene tree, which they say is the correct one or the best one according to their criteria, while we compute the posterior. So when we compare, we look at 4 different percentage values here. First, how often do we use, or how often do our method claim that the tree they pick is the one with the highest posterior? How often do we get the tree that they pick as the tree with the second highest posterior? And how often do we get the tree that they pick as the one with third highest posterior? And another interesting quantity is, of course, how often do we not encounter that tree at all, the tree that SYNERGI picks at all in our posterior? So how often is it outside the posterior? And if you exclude the large families here, the values in the first column are, well, around 60%. Yeah, it's 59 or 60% in all cases. The second and the third, the values are between 3% and 9%. And what's outside is between 20 in this case and 30 in this case. So, it really looks decent. But in the case of large trees, we only get 17%. In only 17% of the cases, we pick their tree as the best, the one with the highest posterior. And in 17, that is, and in 70% of the cases, the tree that SYNERGI picks is outside our posterior there. So we looked a bit closer on that by fixating the SYNERGI tree or the tree that we pick as the one with the highest posterior. And we rerun our program and we look at the unnormalized maximum a posteriori value for what we got. And prime GSR, the tree that prime GSR picked first, had in 90% of the cases a higher maximum a posteriori value, which is, to say that if you believe in our model, it wasn't a computational problem that made so many of the SYNERGI trees lie outside the posterior. On the other hand, if you believe more in their model, which is a bit of an ad hoc model, then this doesn't say so much. What could be going on here is, for instance, that there could be lineage sorting, allele sorting, or even though, so I haven't said it, but SYNERGI relies heavily on gene order data. And what could be going on here is that gene order data suggests something, but if we have allele sorting, another tree could actually be the right one. And even though our model isn't sort of defined for or designed for allele sorting, we could be taking that into account. or it's simply gene duplication. Okay, so this is another comparison, in this case with MrBayes. And what we use here is data from the Yeast gene order browser by Ken Wolf and co-workers. And that data is really solid for some cases where the gene order data strongly suggest a specific gene tree. So we don't compare with SYNERGI here because it would be too easy for SYNERGI. Instead, we compare with MrBayes. So this is sort of a comparison where we can see how much we gain by including or constraining the gene tree by evolving inside a species tree. And it's the whole genome duplication vertex, and the one just above it that we're looking at how often we get that in the gene tree we reconstruct. And we do better than MrBayes. For the pre versus post, we have 90% versus 78%. And for the whole genome duplication, we have 66% versus 35%, which is clearly better. But as I hinted before, if we hadn't had a relaxed clock, we would have done much worse. And we can sort of get a version of our program which has a less relaxed clock by having a very small variance for the distribution that we use to perturb the edge times. So the distribution that gives us the rates. If we have a variance which is this small or factor 10 smaller, well, let's look at this case. It's enough. If we have a really small variance, then we get the right vertex. We reconstruct this vertex in basically only 40% of the cases, while MrBayes does reconstruct it in 78. And here we have 7.6 versus 35. So if we hadn't used a relaxed clock, our results would have been much worse than those obtained by MrBayes. Okay, so let me switch over and talk about lateral gene transfer. This is a little illustration what happens. We have two organisms here. Organism A, which have 3 genes: mustard, yellow, red, and organism B, which have these 3 greenish genes. Now, horizontal gene transfer takes place, which transfer this red gene to organism B. And after that, the result is that we have these 2 organisms here. A remains the same. B has an additional edge. An additional gene, the red one, which it got from A. And as most of you know, the sort of conventional view of a tree of life that in a strict tree way describes the evolution of all organisms there's been serious doubt whether there is such a tree, and many people have suggested other representations like the web of life, etc. So our approach is basically that we believe that there is a tree of life, or at least in many parts of whatever representation you have of the tree of life, of the evolution of life, there are some groups where a tree certainly can be used. And then if you don't have too many lateral gene transfers, you can use a tree, and then you can map the lateral gene transfers onto the species tree and get something like this. A type of realization where we have not only duplications and losses, but also lateral gene transfers like here. Okay. And our approach is, of course, then to define a probabilistic model that generates gene trees given species trees with time. And what we do is a fairly simple variation of the model I already told you about, the gene evolution model. So in this case, we have two probabilistic events that create new genes, namely duplications and transfers, and one event that removes genes, losses. So you can basically view this as the model we had before, but instead, when an event, a gene-creating event occurs, so it should be a duplication. So let me say it like this. We had a birth-death process before, and exactly as before, sometimes it gives rise to duplications. In fact, when a birth occurs according to the birth-death process over an edge, we flip a coin, a bias coin. The bias of the coin depends on the relation between the rates for the duplications and the rates for transfers. And the outcome of that coin determines whether we get a duplication, like here, or if we get a lateral gene transfer, like here. Okay. So we may have things like this that happens during the evolution and continue like this, and then we may get something like this. So again, we have a lateral gene transfer here, and there's lots of duplications here. And we prune away the losses exactly as before, which leaves us with a realization, a different type of realization that contains duplications and lateral gene transfer. And all the vertices of the gene tree have times associated with them. And it's clear that if we would use for a species tree like the one here in yellow and the gene tree like the one on the right here, if we would use this realization to the left, or if we would use this realization to the right, those two realizations would give completely different constraints to the sequence evolution, right? If there's a fairly long time over this edge here, same edge as this, then we would, in this case, expect this gene and this gene to be really similar, while in this case, to the left, the same genes, this one and this one, have an ancestor back here. So we would expect them to be much more dissimilar than in the case to the right. So it's really useful to add sequence evolution to this to get a more powerful machinery. And we do that exactly in the same way as we did before. So now, instead of starting with the gene evolution model, we start with this model that has 3 events: duplications, transfers, and losses. And we use that to generate a gene tree with times on the edges. And then we just continue as before. We generate rates that gives us lengths instead of times. And based on the rates and the gene tree, we generate sequences on the leaves. And that's our generative model. And we can do exactly the same thing as before. Given sequences and a species tree, we can ask for the posterior distribution over gene trees, or we can ask for this posterior distribution over rates. Okay, so I just want to show you a bit of what we can do in this framework. We haven't analyzed any biological data yet. We're really eager to do it, and we have some biological data that we are preparing. But our first priority is always to use synthetic data and check that things are behaving well. And in this case, we have focused on recovering rates. So we have generated from several randomly picked species trees with time, we have generated, as I describe here, we generate several species trees using a birth-death process with realistic rates inferred from biological data. And then for each such species tree that we generate a gene tree, and then based on that gene tree with perturbed edge times, we generate sequences. And then we try to recover the rates we used for duplications, transfers, and losses. And this is how we vary the rates. So we have a death rate called mu, and that's always equal to the total birth rate. And total birth rate, by that I mean the duplication rate delta plus the transfer rate tau. And for every value of total birth rate, we vary the rate of for lateral gene transfer tau between 0 and 100% of the total birth rate in steps of 25. So that gives us 5 data points. I'll show you a plot here. Okay. So this is for total birth rate 0.005. Here we have transfer rate, and it's always the case that this data point corresponds to this data point, this to this, etc. So here we have a transfer rate which is 0, so it's 0% of the 0.005 total birth rate, while we have a transfer rate which is 0.005, so 100% of the 0.005 total birth rate. And then when the transfer rate increase, the duplication rate decrease, etc. Until we're at this data point, which is the same one as here, where we have it all the birth rate on the transfer rate and nothing of the total birth rate on the duplication rate. And the blue line is just the values we use to generate the data. The red line is what we are able to reconstruct on average. So this is over many combinations of species trees and gene trees. And we haven't actually we haven't had the time yet to consider variation, how much, the standard deviation or the standard error or anything like this. So we only have this analysis done right now. The green line is a maximum likelihood estimate, but this doesn't mean that we have a maximum likelihood method, and it certainly doesn't mean that we have a maximum likelihood method that is doing better than our Bayesian method, which is the red line. The green line, the red line is given the species tree and the gene sequences, while the green line is based on the method that is given the actual number of duplications, lateral gene transfers, and losses that's been used in the synthetic generation of the data. It's also given the total length of the gene tree, included losses before we prune it. So the ML is based on data that you don't get in practice, and it's just included because it's sort of a more reasonable thing to compete with than the actual rates used in the generation. Because sometimes sort of random drift gives you data that actually looks like it's been constructed from other rates. And the conclusion is, of course, that for rates of this type, we do very well. Okay, we can basically reconstruct duplication rates and transfer rates. But if we look at the total loss rate that we reconstruct, it isn't as good it should always be point at this line here, but this is the red curve is what we get. The ML estimate is the green, and that's sometimes perfect, but not always. And we haven't really understood why we overestimate as we do. You can see on this data point, but we will look closer at that as soon as possible. Now, higher rates are harder. So if we have a rate which is 0.05, then our estimate is a bit away from the actual values used. But we still have a monotonic increase, so we can distinguish sort of lower rates like here from higher rates like here. Okay, so I don't know about the timing, but I guess it's fairly well timed. I would like to end by mentioning my collaborators on this. So it's two senior guys, Lars Arvestad and also Bengt Sandblad, who now is at Karolinska, the medical university here in Stockholm. There's 3 students. Actually, Ali is no longer a student of mine. He has moved to McGill, where he's a postdoc. And actually, Örjan has also moved on. He's no longer a student here. He's in industry these days. But the final guy here, Joel Sjöstrand, he's a student of mine right now. I would also like to thank Hussein, another student who has helped me a lot in setting up this machinery to give this talk.